Hello and welcome to our first video of Stage 2 Biology. We are beginning with the topic of DNA and proteins, and this is in your subject outline. The way these videos will run is at the beginning of every video, I will have the science understanding. These are the direct parts on your subject outline that SACE is looking for your knowledge and understanding in. So when it comes times for your test and your exam, look at this as a checklist. If there are points where you are not too sure or don't have a good understanding of, then these are parts you should go back or you should be asking clarifying questions in class. Okay, so whether that be after the week of watching the videos, the following week you go, I didn't know a bit about this, that is when you can raise it in class and we can try and answer any misconceptions or just elaborate on that understanding. So for today's video, we are looking at these 4 and a bit main points. Okay, we're looking at what DNA is, how it is held together in our double-stranded molecule. We are comparing eukaryotes and we are comparing prokaryotes. Okay, moving on. So what is DNA? This might be a bit of a refresher for those that have done science previously. But DNA stands for deoxyribonucleic acid. This is a large molecule, and large molecules can be referred to as macromolecules. It is within all living things, and it stores and transmits the genetic information mostly found within the nucleus. So in this cell, if I said, is this a prokaryote or a eukaryote? I would hope you can give me an answer as to which that is. We can talk about that more later as well though. We have our chromosomes. These chromosomes are highly condensed of lots and lots of DNA when it finally gets unwound. Okay, so this information coordinates all cellular activities and functions the same way in all living things. DNA is unique as it can self-replicate. All right, getting into the nitty-gritty of it. Let's look at the structure. So DNA is comprised of 2 polynucleotides. So polynucleotides is a combination of a few things. Okay, and these wrap around an imaginary axis, forming a double helix. This gives DNA that twisted ladder-like shape. Okay, and so when it's not twisted, we can have it looking like this in the middle, and it kind of looks like a ladder, with the bases being the rungs, and the sugar phosphate backbone being the outside of the ladder rail. So we call the sides of the DNA the strands, and it runs anti-parallel. This anti-parallel will be important later on, but for now just know that it does run anti-parallel. So the nucleotides are made of 3 key components. We have our phosphate group on the outside, and we have our sugar. This sugar is deoxyribose in DNA. And then we have our base. These nitrogenous bases, there are 4. We have adenine, which has the letter A, thymine with the letter T, and cytosine with the letter C, and guanine with the letter G. We'll talk about how these pair later on as well. But for our backbone, we have our sugar and our phosphate molecule. That is why we call it a sugar phosphate backbone. So here's what that would look like in more of a chemistry lens, looking at the compounds that make it. So our phosphate, that P is where we have our phosphate group. In blue, consistent with the previous picture, is our sugar, it is deoxyribose, and our nitrogenous base. Now, I am not expecting you to memorize these images or these compounds, but you do need to know that there is a phosphate group, the sugar, deoxyribose, which makes the backbone, and the nitrogenous bases. Okay, just looking at that from another picture again, we can see that on the left and the right, we have our sugar phosphate backbones with our sugar molecule and our phosphates. One component of this is referred to as the nucleotide. So that is the phosphate, the sugar, and the nitrogenous base. If we are looking at just the nitrogenous base, that component of the DNA is referred to as the nucleic acid. The bases are the specific ones that are joined. Okay. And so when we get a polynucleotide, that is referring to multiple of these. All right. In the middle, something that wasn't on the previous images was that these are hydrogen bonds. That is what these base pairs are bonded through. Without those hydrogen bonds, they would just be free-floating bits and pieces within the nucleus. The hydrogen bonds keep them together. Now, these are relatively weak bonds, and that's important because DNA regularly unzips for when it is undergoing replication. Okay, so it is purposefully done to have these hydrogen bonds so they are weak and it can easily be broken and reformed. All right, now don't have to memorize what these look like, but I just wanted to show you what these bases do look like. So if you want, as a bit of a challenge, you should go back to the previous slides and try and figure out which one of these bases was shown in the image. Okay. So moving on to our complementary pairing. So within the double helix shape of DNA, we have our 4 bases: adenine, thymine, guanine, and cytosine. Adenine must bond with thymine and vice versa. So it doesn't matter which way you go, you will have adenine bonded to thymine or thymine bonded to adenine, and the same can be said with guanine and cytosine. So if we have a look at our image over here, we can see that we have adenine bonded with hydrogen bonds in the middle to our thymine molecule, or our nucleic acid. On this image, we can also see our sugar phosphate backbone. Our phosphates are the P's. They will always be smaller than the sugar molecules, deoxyribose in this case. We can see that adenine and thymine are bonded here, but if we go down further, we can see that thymine is on the other side of the strand and adenine has swapped. Okay, this will depend on the genetic makeup as to what these bases will be. And we can see the same thing with guanine and cytosine. They have just been flipped up here because they can be done vice versa. Something else to note on this is that we have this 5' end. That is what the little dash there means. It just means prime, so 5' end. And we have our 3' end. Okay. At the bottom, we see 3' on this side, and we see 5' on this side. This is important later on, and when we're talking about that anti-parallel structure, we can see that it runs 5' to 3' here, going that way, and on this side, it runs 5' to 3' going that way. It will always run 5' to 3', but we'll talk about that later in more detail. Okay, a question for you. I have one strand of my DNA. I've got 1/2 of it, and the bases are as follows: ATG, CTTGG, ACTG. Okay, A being adenine, T being thymine, G being guanine, and C being cytosine. Okay, what would the other side of this DNA look like? So remember, it will swap later on, and it is all done in vice versa. So A should be paired with T, T should be paired with A, G should be paired with C, and C should be paired with G. Okay? And so on and so on. Okay? It doesn't matter what side you have, but you need to pair it with its complementary base. Okay? All right, let's unpack some of these chromosomes as they are filled with lots of DNA that has been tightly packed together. So DNA molecules are incredibly long. If we were to stretch them out, they would go for a very, very long distance. And that is because one strand of DNA has got anywhere between thousands to millions of base pairs. Cells arrange this DNA into one or more chromosomes, and that is important later on. These chromosomes are highly condensed, single DNA molecules. They may have associated proteins, and when we're looking at eukaryotes, they have 2 or more linear chromosomes. Okay, more on that later. These chromosomes are only visible in cellular division. So if we're looking at this picture, we can see our cell, we can see our nucleus. In this nucleus, we have our chromosomes. They won't always appear as this X structure. And when we enlarge in this, we can see that somewhere from this chromosome, something must happen that when it does become unwound, it is DNA. Okay. So let's have a look at that a little bit more. All right, I've got this in red because we are specifically talking about eukaryotes. Okay, that's why the previous one was in red as well. I'm just talking about eukaryotes. So each eukaryotic chromosome has chromatin. This chromatin is slightly less condensed when it becomes unwound. So the chromosome is DNA in its most condensed form, and this chromatin contains a mixture of one long DNA strand and proteins. These proteins are called histones, and they allow the DNA to coil around them. This reduces the overall length so that it can fit into the nucleus. So, we start off with our chromosome, which is highly condensed. If we start to unwind that strand from the chromosome, we get our chromatin fiber. This is still lots and lots of DNA that has been wound together, but the way it is wound is with proteins. So if we go down, we can start to see the structure of the chromatin fiber. It is made up of these nucleosomes, which, not to sound confusing, these nucleosomes are just the protein. So histones, these ball-like structures, the proteins are histones. And then we have our DNA that has been wrapped around these proteins. That is what we call our nucleosomes. When we have a lot of these nucleosomes all sort of stacked together and it's a lot more condensed, we get our chromatin fiber. When we have our chromatin fiber that has been condensed and wrapped around with each other, we get our chromosomes. Once all finally unwound, we get our DNA, we have our double helix structure. Okay, so we have our DNA, the DNA is wrapped around these histones. These histones are the proteins. We then get these structures that are called nucleosomes. When the nucleosomes are then condensed more, we get our chromatin fiber. Chromatin fiber then makes our chromosomes. Alrighty. So I mentioned that it is only during cell division that we can actually see these. So on the screen right now are pictures of what it would look like. Very hard to see under your microscopes. So the chromatin condenses. The chromosomes become thick enough that they are visible under the microscope. So if we are looking at these images, these are cells dividing. We can see that the ones that are thicker are where these chromosomes are dividing through replication. They've condensed a lot, and it's only then that we can see them. Okay, so if we're trying to identify the pictures that have it, we can say we can see the chromosomes within these images. Okay. However, if we were to look at something like these ones, we could not say that we see chromosomes. Okay, it is not condensed enough for us to be able to identify it. Okay. All right, we're now still only talking about eukaryotes. So every eukaryotic species, so humans, flies, cattle, insects, plants, these all fall under eukaryotes, have a characteristic number of chromosomes. So in humans, that is 23 pairs. For a fly, that is 6 pairs. Okay, and that could be different depending on what you're looking at. So these are displayed within something called a karyotype. Okay, this is our karyotype. That's what it is showing. It is all our chromosomes. All right, and so depending on the organism, it will have a different number of chromosomes. For humans, it is 23 pairs. 23 pairs, that means individually it is 46 chromosomes. Okay, and we can see that in this image where we've got all these funny looking zigzags. They are our chromosomes. All up, 23 pairs. You'll notice that on the 23rd pair for humans, it doesn't have the number 23, but instead they've shown it with letters. We'll talk about that in a second. And same with the flies. Okay, they've got 2 X's here and an XY there. All right, so the reason that we have that 23rd pair in a different color is because that is actually our sex chromosomes. That determines biologically whether you will be a male or a female and the other things that come along with that. Okay, whereas all the other chromosomes, so 1 through 22 for a human, they are your autosomes. They contain the information that code for somatic characteristics. Somatic cells are just your body cells that are not linked to your sex. So things like your hair, your nail length, your eye color, these are related to your body cells. Okay, your sex chromosomes come from your parents, 1 from each, and that is what gives us a bit of genetic diversity. Okay. Moving on to our prokaryotes, we're almost done. So it's important to note that for a prokaryote, they have a single circular chromosome. Whereas humans, we had all these pairs of chromosomes that were linear. Prokaryotes have a single circular chromosome, and I'll show you a picture of what that can look like soon. This is located in the cytosol, and this is the fluid that fills the cytoplasm. Okay? I'd ask you, why is it not found within the nucleus? Okay, we should know from our prior understanding why it's not, but I'll talk about that in class as well, and I'll probably give you a little hint towards the end of the video. So, the chromosome is unbound, so it is not associated with proteins, whereas in the eukaryotes, it was found in the nucleus and had to be all tightly wound. That is not the case for the prokaryotes. The region that we say it is in is called the nucleoid region. Okay, and this can be found within the mitochondria and chloroplasts, which does suggest that it may have come from another organism through evolution, and we get to talk about that later in our evolution topic. So, I'm not a liar. Here are our pictures. We have our prokaryotic DNA. We see our single circular strand that is attached to itself. Whereas we look at our eukaryotic DNA and we see that it has a start point, and whilst it is all coiled and quite messy, if we were to stretch this out, it would be a strand that is linear. Okay, it does have an endpoint as well. It would overlap, it would do lots and lots of things, but if we were to stretch it out, it would be a single linear strand. So just in summary, to compare the prokaryotic and the eukaryotic chromosomes, prokaryotic are single circular strand, whereas eukaryotes are double linear strands. And these run anti-parallel. We'll talk about that more later. There is 1 chromosome present within the prokaryotic. However, there is 2 or more chromosomes for the eukaryotic. They are unbound in prokaryotes, so that means they do not have those proteins. They are all loose, whereas in the eukaryotes, they are bound to proteins, and these proteins are called histones. Within the eukaryote, they are found within the nucleus. However, in the prokaryotic, they are found in the cytosol. And where we say it is in the cytosol, it is called the nucleoid region. So in summary, we have looked at DNA stores and transmits genetic information. It functions in the same way in all living things. DNA is helical, double-stranded molecule. It has our sugar phosphate backbone and our nitrogenous bases. In eukaryotes, DNA is bound to proteins. These proteins are called histones. I hope you're sick of hearing me say histones and you remember what they are. And they are linear, which are found within the nucleus. DNA is unbound and circular in the cytosol for prokaryotes and in the mitochondria and chloroplasts of eukaryotes. So they have their own DNA that is separate from the DNA found within the nucleus. Okay, and we've compared the chromosomes in both prokaryotes and eukaryotes. So in class, there will also be supplementary material. We will go over other things in more detail with questions, hoping that you have an understanding of these concepts ready for discussions and other activities. There'll be videos also on the Google Classroom for further understanding and practice material if you want questions for things like the exam and tests.